Welcome to Science Easy Tech channel. In this video, we are going to discuss about microbiology, types of vaccine, 5 marks answer key. This video will be useful for Bsc Nursing students, third semester as well as post basic Bsc Nursing students. Before moving on to the topic, if you are new to Science Easy Tech channel, just take a moment to subscribe our channel and also to press the notification bell icon in order to get connected with our latest updates. Let's move on to the topic. A vaccine. A vaccine is a biological preparation that improves immunity to a particular disease. So a vaccine, you are preparing it biologically. Why you are preparing this vaccine? In order to improve the immunity for a particular disease. The vaccine typically contains an agent that resembles a disease-causing organism. So the agent can be a bacteria or a virus in a weak state or inactivated killed state. So this will be useful to produce an antibody response in the host. So a vaccine typically contains an agent that resembles a disease-causing organism, microorganism, and is often made from weakened or killed forms of the microbes. So history, during the late 1760s, there was a surgeon, Edward Jenner. He has identified vaccine for smallpox. So how he has identified this vaccine is, he has identified the vaccine from cowpox. So many were dying due to smallpox infection, and he has identified this vaccine first. So in 1796, Jenner took pus from the hand of a milkmaid with cowpox, scratched it into the arm of a 8-year-old boy. 6 weeks later, inoculated the boy with smallpox, afterwards observing that he did not catch any smallpox. So in this way, he has reported in 1798 that the smallpox vaccine was safe for both children and adults. Because of the smallpox vaccination only, today smallpox has been eradicated from the whole world. The second generation vaccines was introduced in 1880 by Louis Pasteur, who developed vaccines for chickenpox, cholera and anthrax. So the second generation vaccines was introduced in 1880 by whom? By Louis Pasteur. Louis Pasteur developed vaccines for what? He developed vaccines for chickenpox, cholera and anthrax. So from the late 19th century, vaccines were considered a matter of national prestige, and many countries started developing vaccines. So what are the different types of vaccines? You have so many types: live attenuated vaccine, inactivated vaccine, subunit vaccine, toxoid vaccine, conjugate vaccine, DNA vaccine, and recombinant vector vaccines. So live attenuated vaccine, inactivated or killed vaccine, it is otherwise called as killed vaccine, subunit vaccine, toxoid vaccine, conjugate vaccine, DNA vaccine, recombinant vector vaccines. We will see one by one. Live attenuated vaccines contain a version of the living microorganisms, but it will be in very weak state or in very minor dose, which can produce only antibodies but not infection. But even this live attenuated vaccine, you should not give for patients with HIV or patients on chemotherapy and patients who are on immunosuppressants like that, and all. For them and all, you should not give. Okay. So examples of live attenuated vaccines are measles, mumps, chickenpox, then BCG vaccine, oral polio vaccine. So these are all live attenuated vaccines. Next we will move on to inactivated vaccines or killed vaccines. So here the organism is destroyed, that is the disease producing organism is destroyed by chemicals, heat or radiation. So these vaccines are safer because it will not produce disease, it will produce only antibodies. Example, influenza, polio killed vaccine. Oral polio vaccine is live attenuated. One more vaccine which you will be giving is killed vaccine, that is injection form you will be giving. So that is called as Salk vaccine. Killed polio vaccine is called as Salk, SALK, whereas oral polio vaccine is called as Sabin, SABIN. So hepatitis A and rabies vaccines, these are all killed vaccines. Next is subunit vaccine. Instead of the entire microbe, subunit vaccines contain only the antigens that best stimulate the immune system. No need to give or the whole microorganism. Instead of that, the toxin producing, the toxicity is there no in the bacteria or virus, so that the toxin producing substance antigen alone you will be giving in this vaccine. So in some cases these vaccines use epitopes, the very specific part of the antigen that will be promoting antibody development or T cell recognition. So because subunit vaccines contain only the essential antigens, so better immunity will be produced against that particular organism. For example, plague immunization. Next is toxoid vaccines. For bacteria that secrete toxins or harmful chemicals, a toxoid vaccine will be the answer. So these vaccines are used when bacterial toxin, for example tetanus toxoid and diphtheria toxoid. So all these vaccines can be produced. Such detoxified toxins called toxoids are safe for use in vaccines. Example is, as I have told, tetanus toxoid vaccine. Then one more vaccine is diphtheria toxoid vaccine. Next is DNA vaccines. So DNA vaccines, now in experiment they are doing DNA vaccines, so which will be affecting the bacterial DNA or viral DNA like that, and also the genetic material will be altered. So example is influenza vaccine. Next, recombinant vector vaccine. So recombinant vector vaccines are experimental vaccines similar to DNA vaccines. So they are also like more or less related to DNA vaccines. So but they use an attenuated virus, that is weak virus or bacteria, to introduce microbial DNA to cells of the body. So here we are introducing attenuated, means weak virus or bacteria which is introduced. So this microbial DNA to cells of the body. Vector refers to the virus or bacteria used as the carrier. Example, DPT vaccine. Hope this video is clear for you all. If you like my video, don't forget to give a thumbs up, like and share and subscribe to Science Easy Tech channel. My previous videos I have given in description box, suggested end card and iCard, or you can watch our channel playlist for more nursing related videos. Thank you friends. Keep supporting to Science Easy Tech channel.